Hi, my name is Lemma.
I'm Bezia.
Hi, my name is Lemma.
Hi, my name is Linnea. Kill me. I'm trying.
Hi, my name is Linnea.
And I'm Bezia.
And this is the Lemma Journal Club.
You don't want to say my joke.
I don't understand your joke. I thought it was another typo.
No, it was a joke. And then
hi, my name is Linnea.
And I'm Bezia.
And I'm Linnea. And this is the Lemma Journal Club.
What is the Lemma Journal Club?
Journal Club is a show where we tagline here.
What's the tagline going to be?
A Journal Club is a show where we read papers and talk about them, shit on them.
No, try not to shit on them. Try to say creative things.
Try to be positive and learn something new.
Today, we are talking about the research paper Cellular Hallmarks from Volume Electron Microscopy reveal developmental progression of Plasmodium ookinetes. Do you know how to say ookinetes?
Ookinetes.
Is that correct?
No, I assume it's like oocytes. Ookinetes.
Ookinetes. Oocytes. Okay, ookinetes. So now my notes say the non-picker needs to summarize the paper. And so there's this little gray box that you could now read because you didn't pick the paper.
That's a long thing to read. Okay.
Well, you could try to make it exciting. I'll insert little sound effects and stuff.
Okay, so this paper, I didn't actually see who it was from. It's from University of Heidelberg. Nedal Darif is the first author, and Yannick, Schwab, and Friedrich are the last authors. And the blurb about this paper. Blurb. Unicellular organisms or cells of metazoans often change their morphology during development or life cycle progression to adapt to environmental changes. Malaria parasites undergo a striking range of morphological transformations as they navigate through the different environments of mammalian hosts and mosquito vectors.
Wow.
These developmental transitions are accompanied by changes in the subcellular organelles. Here, this work introduces an unbiased approach using volume electron microscopy to facilitate cluster analyses of morphometric parameters during developmental transformation. Investigating the transformation of fertilized Plasmodium zygotes into the motile ookinetes with 3 complementary VEM techniques reveals intimate mitochondria nucleus interactions, different microtubule arrangements, elongated shapes of the micronemes, and their close interaction with the apicoplast. Woohoo! Our favorite organelle mystery.
No, wrong.
Oh, not our favorite. My favorite. The presented data and approach provide an open access subcellular atlas for ookinete development to aid mechanistic molecular insights from reverse genetic studies and a framework for the ultrastructural study of other parasite stages and developmental transitions in general. I think our introduction
boring
needs
as shit. Jesus Christ.
Improvement.
All right, so we got to find a different way to do that. Normally these blurbs are like 3 sentences long.
Well, this is 3 sentences.
Oh, really?
Well, 4 sentences.
God.
Yeah, it's just a few very long sentences.
All right, so now the format says after the read.
After the read. Oh, so since I was not the picker, I was the pickee. Now I ask you, why, as the picker, did you pick this paper, Bezia?
I picked this paper because we have been talking about malaria, and specifically we've been talking about mitochondria, which I like, and apicoplasts, which you like, and how they might interact. We've been searching for mitochondria apicoplast papers for a while now. We've read a couple of them in test recordings of this podcast, and this is the last man standing. This is the only paper left that we haven't done on this topic. So that's why I picked it. I actually began reading through it. I had quite an arc in reading through this paper. I began reading through it really excited, because the data is really exciting. It's really this time about mitochondria and apicoplasts in malaria. It's about the thing that we've been talking about rather than something kind of tangential, which is what all the other papers were about. And then I got really depressed because this paper on first pass is pretty boring. It's a pretty boring paper. It's a pretty nothing paper. Sorry for the people who wrote it. You did an incredible job, because then my arc went up And I really decided that I loved this paper. There's some really incredible things in this paper. I just have some issues with it. But I actually ended the paper being like, oh, I think this will be kind of exciting to talk about.
Oh, great. I'm glad you ended on love. I think I ended on mid. That I had a low point where I thought that this was quantitative biology at its worst. But I felt that the beautiful images really rescued that low point for me.
I do, for the most part, feel this is, as you said, quantitative biology at its worst. But I decided not to view it that way. So the thing that made me view it as quantitative biology at all is that they do this, they've got these shapes and they try to do some like principal component analysis to say, how do these shapes change and can we create some sort of pseudo time? And I thought that this was really silly,
Yeah. Also, they only have 28 cells. I was confused why you were, I guess it's to make it unbiased that they're trying to get a computer to do this.
Okay, so here's my thought about why this is not so silly, which is that we are used to being able to time resolve our data. Mm-hmm. And so we can see, one cell turn into two cells, turn into an egg. We can watch things happen. They have 28 pictures, just 4x6 glossy photos that they took. And they have no idea what order they go in, right? So they've got they went to a football game, but they weren't able to record the football game or even watch the football game. And instead they took 28 pictures of the football game. They brought them home and they're like, okay, let's figure out how football works, right? It's totally useless, right? It's not totally useless, right? So what they say is, we don't even know what order these pictures were taken in. And it's not clear, because we don't know the rules of the game, that we can guess it. So let's at least come up with a mathematical way to order the pictures so that we can intelligently talk about anything here.
Yeah.
And from that perspective, I actually think that this principal component analysis, to take a bunch of shapes and try to order them in time, is a pretty cool idea.
Okay.
Yeah. So I think that we just came in with a strong bias of hey, just temporally resolve what's going on, get more pictures, make a movie. And they go into the discussion about why that wasn't possible for them. They say, look, there's throughput issues here.
Yeah. I do know that this is very labor intensive. Like the people in my lab who do similar techniques, it's you taking the picture is not easy in and of itself, but then it's once you have the picture, you have to segment it, and that is currently still a tremendous process, even with emerging AI tools.
Yeah. So essentially, I am going to say you should speak louder. It's my guess.
I should speak louder.
I think so.
Okay. Anyways, I'll try to speak louder. Aren't you worried that your microphone will pick me up?
Well, I'm looking at your audio file right now. Who's louder on your audio file, me or you?
I just am trying to keep my paper up, and so my audio file, I need to have a second monitor next time we do this.
Yeah, that's a good point. I have to pull. I'm giving you shit for not having your audio file up, and I also do not have my audio file up.
I'm louder.
Okay.
You're still quieter. I mean, you're bumping a little bit, but you're not bumping a lot.
All right. We'll see if bumping a little bit or bumping a lot works. For those of you confused, we've lost the last few recordings because I'm just like an echo ghost on Linea's track, and we haven't been able to solve that yet. Okay, but I think with this perspective for figure 2, where they've taken these 28 pictures and they've clustered them by random geometry, right? Doing principal component analysis where it normally doesn't belong, in geometry rather than in genetics. And then they try to separate these out into time points. They go to a big hoobah about unbiased, ooh, look at us fancy, which is maybe not entirely deserved and not a best way to sell what they're doing, right? It's really pulling language from genetics. But if you pull it away from that language and think, oh yeah, if you've got 28 pictures of football and you're like, man, I really don't know, are they moving up and down the field with time? Can I use just their x-axis on the field, their yardage as time? And the answer is no, right?
So was that really unknown? That wasn't clear to me that this, that they had no information about what the morphology would look like.
I think they have rough information about what the morphology would look like. Right, and they kind of show this in B, right, where they show that goes gametes and then zygote, and then it forms the retort, and then it makes like a banana that they call the ookinete. Ookinete from oocyte, we're going to say. It's the same double O. So I think they have a rough idea. But I think the hope is like, if you've got something between stage 5 and stage 6, how do you tell if stage 5 is before or after stage 6? They both look like bananas attached to, a sphere. Is the generous interpretation of this shape. Yeah. And so is it more banana, less sphere, or more sphere, less banana?
But why do we care about putting them in order?
Well, so then they're going to go on later in the paper to talk about how the mitochondria move, how the nucleus move, how the apicoplasts move, and being able to say, look, the mitochondria are moving from the sphere into the banana over time is a nice thing to be able to say if it's true, right? Mm-hmm. And they can't say that. They never have any of their x-axes as time. They always have their x-axes as these stages that they made up, and they made their stages up in this quantitative biology way. So this is my forgiveness. This is my explanation of why I think their staging and their principal component analysis and their hoopla is justified. Have I convinced you?
I'll again just say mid-convinced. I'm not up on the literature of what is known about this system, and so if it's really unknown that you go from this blob with a other blob attached to this extended blob with a tail to this total elongated cylinder, then I buy the football analogy. But I was under the impression that the general arc was kind of known, and that here the novelty was supposed to be in the subcellular resolution.
I think the general arc is known, but if you go to figure 3, there's like a stage 4 and a stage 5. One can intuit qualitatively, hey, these are slightly different shapes. But if you imagine that you have several cells at stage 4 and several cells at stage 5, that the variation between cells is going to be bigger than the variation between those two stages, right? So how would you temporally order cells that are at stage 4 and stage 5?
I mean, how do you temporally order them? Like, look at figure 2D and look at stage 4 and stage 5 cells.
Right, they look pretty similar. And so they're saying, hey, look, from principal component analysis, we feel that we can separate these out.
I mean, even if you look at their spectral seriation value, like, this is a tough line to draw.
Yeah, it is a tough line to draw. I agree. But yeah, if you want to talk about mitochondrial migration into the banana, which they have a better word for, they are so heavy on acronyms in this paper.
SBF, SEM.
Yeah, anyways, it makes it hard to remember what anything is called. But there's a word for the banana, and you could imagine that wanting to study migration to the banana is important.
Sure. And if that's happening at the transition from 4 to 5, then I see why they separated them out. But.
The other thing that they say that I wish I understood, they said upon passage to a mosquito host, the parasite's metabolism transitioned from glycolysis to oxidative phosphorylation, rendering mitochondria a key organelle of interest. Is that the time period that we're looking at? Are we now in the host and the metabolism is switching to oxidative phosphorylation? I'm kind of lost on where we are.
Yeah,
what creature are we in right now?
I was definitely lost on where we were. In terms of their figure 1A is look at this life cycle, this beautiful complex life cycle of this organism. And
I think we're at the start of 1A and that we're in the mosquito.
We're in this blue halo, I think.
Yeah.
This gametes to ookinete.
Which looks like it's on the mosquito side. And I think we're transitioning from glycolysis to oxidative phosphorylation.
For the mitochondria.
Yeah, and that we're now doing respiration.
Cool.
And that's kind of one of the transitions. Oh, the banana is called a retort.
Retort.
Yeah.
Yeah.
That's a word. Okay, quick quiz. I'm going to read out a sentence and you're going to describe what the acronyms are. While SBF, SEM.
I don't know what SBF is.
You don't know what SBF is? What is SEM?
Scanning electron microscopy.
Okay. Well, SBF, SEM, readily revealed nuclei. FIB, SEM. Do you know FIB?
So FibSim is where they cut as you go along.
Yeah, I think it's focused ion beam.
Yeah.
Well, SBF, SEM readily revealed nucleoli. FibSim also allowed the detection of CPs. Do you know what CPs are?
CPs. I don't remember what CPs are, no.
Centriolar plaques.
Okay.
I don't know what that is either. They also allowed the detection of SEETs. Do you know what SSETs are?
No.
In addition, revealed microtubules. We observed 4 CPs per nucleus arising at stage 7. Intranuclear microtubules, INMTs, they introduced a new one right there, emerged from the CPs and ranged from 100 nanometers to 800 nanometers in length.
Yeah, this kind of language I just could not, like the gratuitous numbers. Somebody spent a lot of time making those numbers and put them in the paper. It does not help, in my opinion.
Yeah, and I feel like they didn't need, Yes.
That you could have those numbers. Like, it's a beautiful data set. You don't need to report every number that you got. It is not helpful to me.
So did you know about these microtubules in the retort before this paper?
No, I didn't know about retorts before this paper. Did you?
No, right. You had mentioned apicoplasts and mitochondria, and I don't think I knew anything really about malaria before we started this adventure. But I am interested to see our old friend, the micron-long microtubule, coming back.
Yeah.
Makes me excited to think about what one could do.
With the system or with the very long microtubule?
With this system. Okay, so I talked about what I enjoyed about this paper. And then I think we're glossing over, but if anyone's looking at the paper, what's really unbelievable about the paper are these figures 4, 5, and 6 of, segmentation of the nucleus, the mitochondria, and the apicoplasts in these cells over time. There's very little meaningful quantification in the paper. There's very little deep thinking about the paper, but they released these data sets out into the wild, and, they've essentially collected some beautiful data sets and said, look at this, look what you could be doing, right, is what the paper is, and that's why it's beautiful.
So if your question is, what did I find beautiful about this paper?
That is my question, yes.
Yeah.
Linny, what did you find intellectually beautiful about this paper?
I mean, I am fascinated by this transition between stage 6 and stage 7.
Correct.
Where you go from having this little blob with a banana, as we've been referring to it, where the nucleus is in the blob, to all of a sudden you release the blob and the nucleus centers itself in the cell. What does that transition look like? What's going on with the cytoskeleton? Are you moving the nucleus, or is the whole membrane bag moving around the nucleus?
I'm so obsessed with the mitochondria that I didn't even notice that this was the same stage that the nucleus moves into the center of the banana. I was more concerned with that this is the stage that mitochondria migrate from the sphere to the far side of the banana.
Well, so it looks like the mitochondria are migrating, stage earlier.
Yeah, a little bit earlier.
A little bit ahead of the release of the constriction. I mean, I don't really know what's going on that separates out the bulb from the retort, right?
They would like us to be interested in the mitochondria shape, that they feel that these discoid and bowl-like shapes are not really doing oxidative phosphorylation. They also would like us to be interested in the microtubule number, where it's just a mitochondria, and it's attaching to itself or not attaching. I don't think that. Anyway,
the mitochondria number, and also the apicoplast number and stuff.
This
number does not matter
quantitation
here.
Gone bad, right? This is like you have some spaghetti, and it's like, how many spaghetti pieces do I have? When they're overlapping, they only count as one. It's 1, 2. It's 1. It's 2. Right?
Yeah, I mean, I guess it would have been interesting.
I don't think there's any way that number of mitochondria.
I think it could be interesting if it doubled at a particular point to indicate that all of the mitochondria divided. or if there was a maximum size or something but seems like, it's a network of mitochondria that are randomly attached or not attached based off of probably the liquid-like properties of the membrane.
Is there anything that you did not understand about this paper that you want to ask me or maybe our listeners?
Okay, I had a question for you. Let me find it. I highlighted it in green. So I'll just read the statement in the paper, and hopefully the question will come back to my mind.
Do it.
So it says a close interaction of nuclei and mitochondria has been observed in different apicocomplexins in Plasmodium stages. However, the tunneling found in the Ookinetes appears unique. We speculate that it might ensure rapid transport of energy either into the nucleus for meiosis or into the nuclear envelope to enlarge the ER and aid generation of secretory vesicles. So is this established in the field? Because I thought that this notion of intracellular localization of like energy production was pretty new. Is that not true?
That I made it up, yes.
Yes.
Figure 4H, if you look at it, shows these mitochondria essentially tunneling into the core of the nuclei. Maybe people see this normally. Maybe this is not seen normally. I have no idea because I do light microscopy, not electron microscopy.
They said, from that paragraph, they said this is not normally seen.
Certainly is interesting in a lot of ways.
I've seen images, microscopy images of nuclear membranes that have tunnels through them. And I don't think mitochondria were labeled in those cells. So they could have been filled with mitochondria. I don't think that that's the prevailing assumption in the field, though.
Yeah, it certainly is pretty wild to see mitochondria in the nuclei, essentially, or probing into the nuclei. I don't quite understand what needs to be in the why you might need more energy in the nuclei at some point rather than another point. Is it doing like more information processing at one point? Would be a crazy concept that there's it's doing so much information processing in the nuclei that it needs to get mitochondria in there because it's somehow energy limited.
Yeah,
I don't know. I feel like
another thing that I noticed was that they said that the nucleoli arise during a particular stage. I'm going to forget what stage.
What are nucleoli? I know this is a dumb question, but you're the person to ask. They're just dots. The nucleoli are just dots?
Nucleoli are phase-separated liquid-like condensates inside the nucleus that are associated with ribosome biogenesis. So
ribosome biogenesis. So they're just dots. All the other words meant just dots. They're just dots that are associated with ribosome biogenesis. They make ribosomes.
Yeah, they make ribosomes. So
what made them then?
What made them?
Well, the ribosomes are going to make everything else. What made them?
Well, yes.
Yes.
Well, so this is what confused me about the arrival of a nucleolus. What was happening before the nucleolus was there? I know that during cell division in eukaryotic mammalian cells, that the nucleolus dissolves and then recondenses at the nuclear organization regions or whatever they're called, nucleolar organization regions. But that doesn't seem to be what they're talking about here. They're talking about at a particular stage, you can now see the nucleolus. So what was happening before that stage? Were you just relying on the old ribosomes and the that were formed in the previous stage, and those were making all the proteins, and then at some point you need to increase protein production, so the nucleolus starts to emerge? Or if you're making ribosomes at a low enough rate, maybe you don't need a nucleolus. Maybe you just, you're able to do the rRNA processing without the nucleolus.
Yeah, this is why I say this is an exciting paper in the end, is they explain nothing. They do no thinking about what they've gotten. I know very little about biology, so I'm totally lost from the get-go. And what you get out is like a bucket of infinite questions from just like these 28, 4x6 glossy photos that they've taken, that they've arranged in an order that they find pleasing. And so like on that, here's my question for you about this paper. What is figure 7?
Sorry, let me find it again. I zoomed in on figure 4H.
Figure 7B.
Wait, before we move away. Okay.
Sorry.
Never mind. We'll come back to it.
Will we? I think we're getting towards the end. We got to go get Asya soon.
Okay. I was just going to say figure 4H, one of the other things I didn't understand about this, 4G and 4H, is like, why is the nucleus changing shape here? Like it has these little protrusions, and like in 4H, that's sometimes where the mitochondria are localizing, are the like protrusions in the nucleus. And what's going on there? Is that an artifact of the imaging? Could it be an artifact of the imaging or
if
segmentation?
If it was microscopy, I would say yes. I never quite know what to believe about electron microscopy.
I kind of believe it. Right, because that could be like the constriction.
Yeah.
And the nucleus is starting to move before the constriction has been released, I could imagine.
I think happening. A little bit of H, what you're seeing, is the problem of level sets to create a surface. That this is a little bit of a processing, I'm going to go quote unquote artifact.
Yeah.
That, the way that they have to make this is level sets. And so, when there's a second surface, the mitochondria, the level sets are getting a little bit wonky. Is kind of my interpretation, but that could be totally wrong. I don't know. I don't see the raw data. So, and even if I did see the raw data, I can never interpret electron microscopy raw data.
This is so hard to interpret raw. Like the segmentation is a lifesaver here. So,
all right. My question, what is figure 7? Specifically, what is figure 7b? The micronemes
I tried to look up micronemes.
Micronemes are responsible, I figured it out, are responsible for making stuff that is going to help this banana move.
Help the banana move? I thought it was something about
the mitochondria mostly make energy.
Yes.
The apicoplasts mostly make lipids.
Yeah.
And the micronemes mostly make proteins that are helpful in moving. Okay. That is my summary of biology. I can find a quote if you want.
No, it's okay. It's okay. What are you confused about? These are just beautiful images.
What is this apical tubulin ring? What is this? It looks like I'm looking at like ATP synthase or something. There's this beautiful, incredible structure that these microtubules and this wonderful like spindle pattern that outlines the banana. And what am I looking at?
Yeah, I'm not totally sure. The B top view made me think of like flagella. But I don't know what we're looking at.
All right.
I'm sorry.
It's okay.
We could ask our listeners or lack thereof.
Listeners, if you are looking at this paper, figure 7, what is this? And we know, it's a banana with micronemes in it. But more specifically, what does the structure in B really do that we're looking at?
But I love these images. I mean, like the segmentation and
But then it's EM segmentation, and they talk about that they don't use the newest cutting edge techniques to fix the sample, so there is deformation. So like, how much of this, if anything, is real? It's beautiful, but there is always a worry when looking at this kind of data that
but some of it, I mean, like the overall shape has to be real, right?
It has to be real.
You don't think that this bulb with a tiny banana to an independent banana?
Okay, sure. The gross shape that you can also see under light microscopy does have to be real.
Yeah,
I agree with that.
And then I would argue that the nuclear localization also has to be real. And to me, that was the most exciting piece of information in this.
If you could magically wave your pipette and have one experiment added to this paper, what would the experiment be?
Oh, yeah. Time lapse between stage 6 and stage 7.
Time lapse. They don't even talk about it in the discussion. They're like, oh, we could use, expansion microscopy. And they're so given up on normal microscopy because these things are so small.
They might be too, but like you could label the nucleus and just watch the nucleus move, and that would tell you a tremendous amount.
Yeah, same with the mitochondria. Super resolution microscopy. I know it's a pain in the ass, but God, would it be nice to go alongside this data.
Yeah.
And
just
so
a little bit of fluorescence. Yeah.
Can you get us these malaria cells?
We can talk to Josh.
Do you
see if he's
need like
interested.
mosquitoes and like mice? Like they've got this 1A, which shows a mouse and a mosquito, and then maybe like do we need all these creatures in order to be able to culture these guys, or can I just culture these guys? I have a bad feeling that we need mosquitoes and mice, and we have to go to a mosquito lab, which I don't want to do.
I'm not sure that we do, but we can, we can reach out to either Josh, your neighbor, or I met a woman at this conference, the HHMI conference that I went to, who's at Berkeley, who's also doing malaria stuff. So,
any deep takeaways from the paper? Things you felt that you learned?
I mean, I don't know. Yeah, it's nice, label-free. Yeah, advancement of our understanding of biology.
I don't think I care about label-free if you have to fix the sample for electron microscopy.
Oh, yeah? I feel like it gets people interested in systems that aren't genetically tractable yet. And so I, in that respect, and it opens up questions I wouldn't have thought of otherwise.
I just usually don't think about labels as a genetic thing because I'm so into all these dyes. I'm like,
all
I have a tubulin dye. Oh,
Yeah.
I don't have a microneem dye. That's fair, but But just wait, I'll make one.
Yeah. I mean, it does sound like the apicoplasts have multiple membranes. Anyways,
4 membranes. The apicoplasts have 4 membranes.
Yeah.
And then they show a picture of a mitochondria where they're like, oh, it has a double membrane. And what they mean is that it has a double membrane. There's like a mitochondria in a mitochondria.
Oh, and then I just want to highlight, because we didn't even talk about it, and we do have to wrap up pretty soon, or imminently, is the membrane wrinkling figure panel. Did you have any thoughts about that?
Which one is this?
This is 3EF.
No, I flagged it to try to go look stuff up. I have no idea what that is or what I'm supposed to think about that. Is it a mechanical instability?
Is this a mechanical instability, or is
know.
this something that the cell is doing? Yeah, that's the...
No idea.
To have such sharp... Like if you look in the FibSem right panel, it's so sharp.
Yeah, I have nothing. I meant to go try to track it down, but...
Yeah.
All right, so that's about it. We've got to go get Asya. Hopefully you enjoyed the Lemma Journal Club. Let us know if you listened to it and your thoughts about it and how bad of biologists we are. And we'll try to pick something more physicsy next time so that we sound a bit more intelligent. But honestly, we really like reading these biology papers, so it might be a lot of this.
Thanks for listening.
Bye.
Are we going to do a clap out?
Oh, sure.
Okay.
All right. Are you ready?
I'm ready.
So we're going to go 1, 2, 3. 1, 2, 3. It's like giving ourselves a little applause.
Woohoo!
Okay. I'm hitting stop.
Stop.